Today we're going to be talking about something called a Punnett Square. And a Punnett Square is a tool that we use in order to figure out the possibilities of genes and phenotypes for offspring. So the way that we set up a Punnett Square is we're going to make a grid. With a box. And so what we're going to do is we're going to put the father's genes, or one parent's genes, on the top. And then we're going to have the mother's genes, or the other parent's genes, on the side. So if we know which is the father and which is the mother, we can set it up like this. But if we don't know, then honestly it doesn't really matter. You can put either in either position. Now, the other things that we know so far is we know that if I have a capital letter, then we know that that is the dominant trait. And we know that dominant traits only need one of that version of the allele in order for that trait to be what we see. The other thing that we know is that capital letters come first when we're writing a genotype. So they are written first. So an example would be like H, where the capital letter is written first. So let's set up a Punnett Square here. We've got our grid set up, and we're going to put the genotype of the father on the top. And we're going to use H because it's an easy letter for us to see the difference between the capital letter and the lowercase letter. And let's make the mom have two different versions. So she's got one capital letter and one lowercase letter. So how we're going to fill this out is we're going to, in this box, put one of the dad's genes and one of the mom's genes. So mom would have, her H would go here, and then dad's little h, because that's in this column, would go right here. Now for the next one, we've got mom's H is going to go here, and dad's H is going to go right there. Now, is it possible for me to put dad's H first? Yes, but that would be wrong because mom had a capital letter H, so hers is going to come first. But for example, when I fill out this box right here, I've got this H from dad and this H from mom. And then it doesn't really matter which one comes first because they're both the same lowercase letter. So this is from mom, and this is the one that this baby would get from dad. So what we're really showing here is we're showing what would happen with meiosis. We're showing how the mom's and the dad's chromosomes would get split up and the possible egg cells or sperm cells that would be made. Because of this, we can tell both a genotype and a phenotype and the frequencies of these. So if we remember, we'll do a little definition down here. So genotype, that's going to be the combination of alleles. And then the phenotype is going to be the trait. So that's going to be the trait or the characteristic. So in this case, we're going to have H. H is going to represent hind limbs, also known as back legs, or a lack of back legs. Now my genotypes can be big H, big H, big H, or little h. Those are my possibilities. And we know from our drakes that the big H, the dominant one, is having hind limbs or having legs, and then little h would be no legs, no legs. So I could have used L for this, but L is a much harder letter to tell the difference between the capital and the lowercase. So looking at this, we've got our mother and our father. We've got mother is a heterozygote. She has one of each type of allele. And the father is a homozygote. His alleles are the same, but he's a homozygote recessive because he has two recessive alleles. This mother and this father have a 50% chance of having a baby that has hind limbs. And I say that because half of these, if these are the possible babies, half of these have a big H, and the other half are all recessive. So this combination of parents with these genes have a 50-50 chance of having a baby with hind limbs. In their baby. So let's take a look now at what other possibilities we have, and we're going to make Punnett squares for each of them. So I'm going to make a little section here. So one possibility that we could have is we could have a parent who is big H, big H. So we're going to keep using the same things here. So my parent is going to be big H, big H. And it turns out that it doesn't matter what I cross this parent with. It doesn't matter what the other parent's alleles are. They could be big H, they could be big H, or they could be little h. And it doesn't matter because if you have two dominants, then 100% of the offspring means babies, will have the dominant trait. Every single time they're going to have the dominant trait. So let's take a look at how this works using our Punnett square. So this time I'm going to make my Punnett square a little bit smaller. So I'm going to set it up. And I'm going to put my big H genotype. And then let's just choose little h just to see really how this works. Because this parent can only give that dominant trait, every single one of these babies is going to have that dominant trait. Because you only need one dominant trait to show it. So all of the offspring will have that, 100%. Let's look at another example and see another type of distribution for the offspring. So let's see what would happen if we had what's called a heterozygote with a heterozygote. So heterozygote means that you have one of each type of allele. And so we're going to cross a big H little h parent with a big H little h parent. When you do this, you will have the following Punnett square. So let's draw it out. We're going to put the big H, little h on top, and then a big H, little h on the side. Doesn't matter which goes where. Now, drawing this out, I'm going to have big H, and then big H. So this one comes over here to combine with this one. And then we've got this one and this one are going to combine in this box. And then this one and this one are going to combine in this box. And what we can see from this one is that when you have this type of a cross, when both parents have one of each, a dominant and a recessive allele, you're going to end up with 75% will have the dominant trait. And 25% will have the recessive trait. So let's take a look at that. The reason that I'm saying this is if I were to shade in all of the ones that are going to have the dominant allele, 3 of 4 boxes are going to have that. Well, 3 of 4 is 75%. Only 1 of 4 is going to be recessive, and so that's only 25%. So that's how I'm figuring out these percentages. All right, what would happen if we had a heterozygous, so a parent that has big H, little h, and we crossed them with a little h, little h. So let's see if you can figure that one out. So big H, actually I wrote that in the wrong spot. Let's go up a line, we don't need to skip that line. So big H, little h, a heterozygote, is crossed with a little h, little h. So take a minute, do this Punnett Square. I'm going to suggest you do it on a different sheet of paper. Now what you should find with this is you're going to end up with 50% are dominant and 50% are recessive. And I know that because that is actually the cross that we did right at the beginning. So here we've got one heterozygote parent with one recessive parent, and they had a 50-50 chance. So 50% were dominant and 50% were recessive. All right, the other cross that we could possibly have is we could have an all recessive with an all recessive, and this is going to have a 100% chance of having the recessive trait. So if we're looking at our dragon game, or if we're looking at any Punnett Square, we can use this as a tool to see what percentages we would have in our offspring. So we could figure out how many children we would expect this couple to have the dominant trait and to have the recessive trait. But if we have the offspring and we know that 50% of the offspring have the dominant trait and 50% of the offspring have the recessive trait, then we can also go back and we know then what the genotypes of the parents have to be, because that 50-50 split will only happen if one of the parents is a heterozygote, they have a big and a little, and one of the parents is fully recessive. So this is a tool. Anytime you see these frequencies, we automatically can tell what the genotype of the parent is going to be. The last thing I want to cover is if you have a parent and you don't know what their genotype is. So let's say they have hind limbs, but you don't know if they are big H or if they're big H, little h. You are going to want to cross them with a little h parent, because looking at this, yep, I can see all of it. If I don't know which of these they are, if they're big H or big H, little h, if I cross them with a fully recessive parent, if I have them make babies that way, then if they are big H, all of the offspring would have that dominant trait. But if they are a heterozygote and I see any of the babies have the recessive trait, then that's what I would know. So by crossing them with this parent, I can automatically tell if I get any babies with that recessive trait, then they have to be a heterozygote. But if all of the babies have the dominant trait, then they're probably big H.